Austin Jones at the Medical University of South Carolina before transitioning to Rutgers about 4 or 5 years ago now. And in that time, she's been very productive and successful. Amy has published about 15 papers with a number under revision for consideration of publication, and most notably her K-99 grant from NIDA that has funded a lot of her work as she's beginning to transition into the independent phase of her research career. With that, I'll turn it over to Amy to talk to us today about developmental exposure to the organophosphate chlorpyrifos in the rat and implications for addiction and psychiatric disorders. Welcome, Amy.
Thank you. Thank you guys so much for having me. It's a pleasure to talk about this work today. I'm going to basically give you a short timeline of what I'm going to be talking about. As you guys might know, I had this amazing opportunity through the T32 here to start expanding my work on addiction and sex differences in addiction to look at the effects of organophosphate insecticides during development on adulthood addiction and psychiatric phenotypes. So I'm going to talk to you briefly about substance use disorders, how we kind of model them in rodents, and in particular this paradigm that I developed early on in my postdoctoral training, in my first year of my postdoctoral training, that I've used to kind of work on these different circuitries involved in sex differences and addiction, as well as applying it to more recent work looking at the effects of this organophosphate chlorpyrifos. So just briefly to go over substance use disorders, from measures taken in 2017, 19.7 million American adults are battling a substance use disorder. Interestingly, and most notably, this is also comorbid with a number of other psychiatric disorders. Almost nearly half of those with substance use disorders also suffer from a mental health disorder. And essentially, this has been a pretty large fiscal issue, $740 billion annually, mostly due to poor policy choices, loss of workplace productivity, crime-related costs, healthcare expenses. And something that, of note that I recently saw was that during the coronavirus COVID-19 pandemic, drug overdoses have actually started to spike, having an 18% increase over prior years, and we're just in October. Also notably about this is that not only are we getting an increase in the number of drug overdoses, the location that this is occurring is changing. So generally speaking, most drug overdoses occur in urban environments when people are out doing drugs with people. But what's happening is that drug addicts are now taking their drugs to their house and doing it in isolated environments in their home, which is now creating a tighter connection between the home environment and drug context, as well as a lesser likelihood of getting treatment if somebody does overdose due to just the lack of general sociability. So when we're thinking about these different aspects of substance use disorder in people, we start to consider, what are the ways that we can actually identify how to study this in animal models? The difficulty with humans is that there's comorbidity. People are very rarely single drug use individuals. They tend to, if you smoke cigarettes, you drink alcohol. Alcohol tends to be comorbid with most. So one of the things that we want to know is, what are the aspects of substance use disorder that can influence propensity for drug use, right? Pathological drug taking. Once somebody has stopped taking a drug, why do they start up again? How do they start up again? How often does it occur? And how does that lead to overdose? And so we tend to think of this as what we call a persistent, cyclic, or pervasive disorder that involves drug use and drug seeking. And for my work, I have primarily focused on the intensity of relapse, so how much drug seeking, how much drug taking, and the frequency of relapse and duration of abstinence. How often do people go back to their drug of choice? Quality of life changes, relative cost is also something that we tend to consider. What are people willing to give up? Their jobs, their social networks, their family, that sort of thing, and how does that kind of come in? Now that is a little bit more difficult to study in rodents, as we don't tend to take their social circles away from them. That's already gone when we start most of these studies. So instead, we're focusing on the frequency of relapse and duration of abstinence. So in order to do this, we can't really ask an animal to stop taking a drug. We have to kind of force them to do so. So what we did was develop this model that we call the seeking persistence paradigm. It's a paradigm that is modified from other, what's called incubation paradigms. So essentially animals are given, their compound drug cues. So we're gonna do it like this. All right, so they're given their compound drug cues. So they're put in this box here, right? This is their self-administration chamber. we've implanted them with what's called an indwelling jugular catheter. So they have this catheter system that goes into their jugular vein. So we can hook them up to this tether that will intravenously infuse drugs. Now, for my studies, I've used cocaine, but we also can do this with a number of different drugs, including opiates and methamphetamine, and basically any drug that has works best with drugs that have quick half-lives. So they're put in this box every day, and what happens is this little light in the top corner here, it's called the house light, will go on, and that will signal to the animal drug is available. And then the animal will go, and it'll hit this lever. This is called the active lever. And when they depress on the active lever, this light will go on, and a tone will play, and they'll get an infusion of the drug. And this will allow them to self-administer the drug over a 2-hour period. And so when the drug infusion stops, everything turns off, the house light turns off, signaling that there's no drug available for the moment. You can vary this 20-second timeout period usually that we use. And then the house light will go back on, and the system will repeat. So from here, we can stably get animals to self-administer drugs. They'll do so on different scheduling. So one lever press is one infusion, that's an FR1. We go up to FR5, FR10. These are measures of effort responding. And then what we do for the seeking persistence paradigm is after 10 days of this self-administration, we actually take the drug away. So the cue light's gone, the tone is gone, the house light is gone, all of this is gone. The animal's still tethered to what used to give them drug. And they depress on this active lever, what used to give them the drug, and we measure the number of times that they press this lever as a measure of drug-seeking behavior. For the seeking persistence, so this is seeking, this is initial seeking behavior. How much effort do they expend to try to get this drug when it can't be given to them? We then put them into forced abstinence for 2 weeks, and we return them to the paradigm to take a look at whether or not they persistently, even after periods of abstinence, when they shouldn't be going through withdrawal symptoms, particularly not for something like cocaine, whether or not they still seek the drug later. And from here, we continue on and we do what's considered operant extinction, where the animals will basically go back into this paradigm repeatedly until they stop hitting the lever. And so from this, we can get a number of different measures. So what the data looks like in general, right? So when we think about this for just overall data, this is active lever presses here. These are days. This is self-administration. These are male rats here alone. You can see them acquire and slowly gain speed in terms of how many active lever presses, how much drug they're actually taking, until they hit the stabilization point. For males, about 50 lever presses for the dosing paradigm I use. Extinction day 1, so that's this one. When we first take the drug away, you can see this huge spike in active lever pressing, right? They want that drug. And we put them into abstinence for 2 weeks, they come back, and that persistently is present. Now, if we immediately send them back into this the next day, you can see this rapid drop-off. So extinction occurs pretty rapidly in male rats, right? So they don't, once they realize the drug's not going to be there, they're not going to keep looking for it. And so we can measure here both the magnitude of the expression of the drug-seeking behavior, the number of lever presses, as well as its persistence over time. And so what we've done in this paradigm is we've said, okay, so this, your first hypothesis would say that, extinction day 1 might correlate to the amount of drug that they want. And what we find is it actually doesn't at all correlate to the amount of intake. It's independent of intake. It's also independent of the number of lever presses that they do initially during self-administration. And in fact, it's really interesting because this tells us that there's a large scale amount of individual differences that are independent of the amount of drug. So, and you can think about this in terms of humans, right? Someone might prefer to have, 5 drinks of alcohol, and that might be their alcoholism, versus somebody who is not coffee has 20 drinks. The amount of drug preference is not necessarily, the amount of drug taken in preference is not necessarily determine how addicted or how much the addiction vulnerability may be present. So we can see this in this paradigm as well. What we found really interesting about this as well is that we can predict how much seeking behavior they're going to do at this secondary point based on their initial seeking performance. So we hypothesized that if we affect ED1 in some way, right, maybe we can have some kind of long-term component on later drug-seeking behavior. So we can really target this initial point, initial phase of abstinence. Maybe we can have some long-term effects. So how does this apply to then sex differences in addiction? One of the interesting things that we look at in our lab, or that I've looked at primarily, is that there are large-scale sex differences in addiction that are present across a number of different drugs, most prominently shown in stimulants. Women tend to progress more quickly from casual drug use to abuse. They tend to take drugs at higher doses. They have shorter periods of abstinence, meaning that their cycling relapse occurs more rapidly. And what's interesting is that we can recapitulate a number of these in smaller forms across different paradigms for looking at drug addiction, but our seeking persistence paradigm allows us to actually see this really strongly within the same animals over time. So this is looking at females versus males for just the self-administration behavior, the FR1. Females acquire much more rapidly. Females have a higher intake amount. This is really important because, as I showed before, ED1 is independent of cocaine intake, so that would have been a confound, but with this paradigm, we don't have to worry about that. And what's interesting is that on extinction day 1, females tend to seek the drug to a much greater extent than do males. This is persistent over time, and also the number of days following this extinction for them to stop seeking the drug is quite a bit longer in females. It takes up to 14 days, whereas usually it's about 7 in males. So we wanted to then figure out, and we had done this in our paper published in 2017, we looked at a number of different neural substrates that were activated on ED1 in males and females, just try to see what substrates could be possibly involved in driving the seeking behavior, number 1, but most importantly, in driving the sex difference in the seeking behavior. Why are females having this greater activation compared to males? And so we looked at a number of different monoamine targets, and just to summarize quickly, we looked in the dorsal raphe, which is the hotspot for serotonin neurons that are projecting to the forebrain and to the hippocampus. And we see this pretty strong increase. These are home cage animals. So these animals were, they had 10 days of cocaine self-administration. They're in a 24-hour withdrawal, but they're not undergoing extinction. These are the animals on extinction day 1. So on that initial extinction event, when they're rapidly looking for the drug, we have this large activation in neurons, in particularly in serotonin neurons in the dorsal raphe, and that those neurons actually correlate, that neuronal activation actually correlates pretty strongly in both males and females to drug-seeking behavior. We looked also in the locus ceruleus, looking at norepinephrine signaling, and this is where things kind of got interesting for us. We see an increase in norepinephrine neuron activation on ED1 in males and in females. However, it was much stronger in females and only correlated to drug-seeking behavior in females. To make a long story short, we also looked at the dorsal hippocampus and regions around the cortex and found that most likely the hippocampus was a big target region for these to be projecting to, and so for subsequent studies, we looked at specific targeting between different substrates targeting hippocampus specifically. So what we wanted to do is focus in primarily on the locus ceruleus and norepinephrine neurons, and we hypothesized that locus ceruleus norepinephrine may be recruited signaling to the hippocampus in females only to drive that increased behavioral seeking persistence that we saw in females compared to males. So in order to do this, we used DREADs. DREADs are designer receptors that are exclusively activated by designer drugs. They're G-coupled receptors, and what you can do is you can create a construct, a viral construct, with a specific promoter. In our case for this, we use the PRSX8 promoter, which allows us to specifically target norepinephrine neurons when it's implanted into the LC. It has HA tags, so we can stain and make sure it's actually in those neurons. And then we can either use an HM3, human muscarinic receptor 3 or 4 modified receptor, to either to selectively induce a GI-coupled receptor or a GQ-coupled receptor for inhibition and activation respectively. These receptors are targeted or can be activated by clonazepine N-oxide, a designer drug, and so this allows us to selectively target pathways. So what we do is we can inject this DREAD into the LC, and then we can inject the drug into somewhere like the hippocampus, in this case, right? And that'll allow us to just selectively activate or inhibit those projections from one region to another. And this can be done pretty much anywhere in the brain. This is just looking at expression in the LC here, showing that we get this really nice arrangement of the DREADs around the LC neurons. And then in projection, so when we hit the actual hippocampus, we can see that it's at terminal sites as well, because the DREADs are transported to terminals. So we use this to both activate or inhibit the circuit to see if we could actually modify drug-seeking behavior dependent on actually changing the amount of signaling of norepinephrine to the hippocampus itself. So, to start out, we have our model here. So we did this dread injection into the LC. We waited 6 weeks. We started the self-administration chaining. On extinction test 1, they either got, this isn't actually saline, this is artificial cerebrospinal fluid or CNO, clonazepine N oxide, into the hippocampus, and we did the extinction day 1 test. Yeah. All right. So here we have this. So this is the males and females. When we look at the HM40I, right? So remember how we said that norepinephrine didn't seem to be as recruited in males, right? So when we then inhibit norepinephrine in males, we don't seem to have any effects on active lever pressing or 81 type behavior. But when we look at females, this is normal females, just with the control injection, right? They have quite a bit of active lever pressing, drug seeking behavior. But if we inhibit the norepinephrine pathway, that's just a singular specific one, we have almost an entire deficit or removal of that seeking behavior. And when we test them 2 weeks out, we can see that these effects are actually persistent. So even though these animals are only getting 1 injection at this time point here, if we alter this initial drug-seeking behavior, we can have a persistent effect 2 weeks later. So of course our next question was, all right, so we see that normally males aren't recruiting this norepinephrine pathway, but does that mean it's not there? Well, we know that it's there for memory. It's important for memory and other things. So the question was, okay, if we excite it in males, can we actually induce a female-like phenotypic response? And in fact, we can. So if we use the excitatory dread, we can actually get males to start to look more like females just by activating this response, suggesting that this pathologically high level of drug-seeking behavior and persistent drug-seeking behavior might actually be due to engagement and recruitment of the norepinephrine system. So this effect with the HM3DQ is not a persistent effect per se, but just having it be transient is interesting enough to us at this point to say that, okay, we can start to think about how norepinephrine in males and in females might just simply be a circuit that's recruited to enhance pathological drug seeking. So over time, I've done a number of different studies using this particular paradigm over my career so far. And generally speaking, these are things that I've done as part of the K99 first and second aim period, looking not only at dorsal orphate serotonin, but also and locus-serious norepinephrine, but also at specific antagonist systems in this oxytocin, corticotropin releasing factor systemically, and we've essentially created this model of these hotspots that are present in driving sex differences in pathologically high demand, as well as some possible treatment mechanisms that can be used to reduce initial seeking during abstinence. But one of the biggest take-home messages that we got is that we have this situation in which female excessive high demand for drugs, or rather high seeking behavior for drugs, seems to be, occurring in a sex-specific manner in terms of recruitment of neural circuits. And in particular, what we've really been focusing in on is this locus coeruleus norepinephrine mechanism. Now this work got us really interested, combined with the opportunity to work on the T32 in this department, this question of, this is what's happening in the normative circuit, right, in males and females. So what if we start to engage mechanisms that alter during development how norepinephrine may function and how a number of these different brain systems may function? Can we start to get aberrant behaviors or changes to the norepinephrine system that have long-term effects? And given the opportunity that we had that otherwise might not have presented itself, we became interested in chlorpyrifos as a possible toxicological agent that might have an effect in the noradrenergic system and thereby influence addiction and psychiatric phenotypes. So to start out, what is chlorpyrifos? So it's an organophosphate insecticide. You guys have probably seen it quite a bit in the news lately. We recently had a pretty big win where California banned it, given its large-scale links to negative adverse side effects in offspring and in children. One of the interesting things about chlorpyrifos is that even with very low levels of exposure, you can have kind of induced developmental delays in adults, and also the fact that the developmental effects are so much more substantial than the effects in adulthood, meaning the same amount administered to a pregnant dam you'll actually get higher levels of it circulating in the offspring than you will necessarily in the dam themselves. So it seems to be that it is particularly important when you're thinking about developmental outcomes. The mechanisms for its action are pretty well identified. It's an acetylcholinesterase inhibitor, which will then thereby increase levels of acetylcholine at synapses. The developmental exposure of it can induce long-term effects. Jason Richardson here did a lot of this work during his PhD on this. And these effects are not just long-term, but they seem to be pervasive, meaning they seem to get worse over time. And so, this occurs not just at with hit it with a hammer kind of scenarios. This occurs at like small interval dosing, very low dosing, but the effects seem to be kind of aberrant, meaning like depending on the dose, you can get either high or low effects. But the point overall is simply that this toxicant can alter acetylcholine during development in one way or another, depending on what the dose is, and that will have long-term effects on brain development and brain function. Now, what we became really interested in is this idea that it's not just acetylcholine that's altered. If you look at norepinephrine, you actually can see that at 1 mg per kg or 5 mg per kg, 1 is blue, 5 is black, you can have large-scale changes in the hippocampus, which has been a hotspot for us, as well as cerebral cortex, and this is, I believe this is amount and turnover rate of norepinephrine in these different regions, suggesting that norepinephrine may be, or at least norepinephrine signaling may be substantially altered by chlorpyrifos in development, even at low doses of 1 and 5 mg per kg. We've even looked at, or people have even looked at previously beta-adrenergic receptor binding, so this is the primary ligand, or the primary receptor site for norepinephrine signaling, and it's been shown that there is quite a bit of cortical changes that can occur, both in male and female rodents, although in different directions. That these effects are not just in beta-adrenergic receptor binding, but also in straight norepinephrine concentration in the cerebral cortex, as well as these have shown to have some behavioral actions in rodents, looking at learning. This is T-maze, so it's basically a decision-making paradigm where an animal either has to change their mind as to go left or right, or they have to, perseverate and continue going right. And what this is actually showing here is that if they've had 5 days of developmental exposure of CPF, they actually have a 0% linear improvement, suggesting that they are perseverating in whichever behavior that they're choosing. So that is, to me, showed a really strong indication that norepinephrine is substantially altered in these animals. And then similarly, they looked at, there's another study that was looking at prepulse inhibition, which is also a noradrenergic derivative response. So our hypothesis was that chlorpyrifos, given its nature as a cholinergic disruptor, could be altering norepinephrine signaling during development, and that would persist into adulthood. We primarily thought that prelimbic cortex might be affected, something that hadn't been looked at previously, as most of these studies have not delineated between different subregions of the cortex as much as we might have liked. And looking at norepinephrine innervation there, we know from prior studies that altered norepinephrine can induce compulsive drug-taking behavior. And particularly things that we've seen before is looking at CV persistence, like I recently just showed you guys. So CPF also, we think, would alter behaviors that require normative norepinephrine signaling to regulate disengage. So this idea that they have to make a decision, their same result is not giving them what they want, they have to disengage from that pathway and go to the other. And we know that that is a very strongly regulated behavior by norepinephrine. So what we did was we looked at, we gave chlorpyrifos in safflower oil on vanilla wafers once daily to pregnant dams from gestational day 6 to 20. This is actually a really cool trick that Jason Richardson told me about, where you don't have to actually oral gavage the animals, they will eat their vanilla wafers. It's pretty fantastic. And so we did this for gestational day 6 to 20. The animals grew up. We stacked a small group of them on PND 21, just to look at hormonal changes, and then we stacked another group of them at PND 60, just to kind of be comparable to where our behavioral results were. In an additional 2 group of animals, we took one group and we did self-administration chaining. For the purpose of today's talk, I'm not going to go too much into the reinstatement paradigms, although I do have that data if you guys are interested afterwards. And And in a second group of animals, we looked at more psychiatric type indices of habituated and non-habituated behaviors. So to start, we first looked at what noradrenergic fiber innervation looked like in different areas of the cortex. So this included, cingulate gyrus, prelimbic cortex, infralimbic cortex, and the dorsoplandicular cortex. And each of these different subregions has a pretty strong impact on decision making, and in particular on drug-seeking behavior as well. So we know that prefrontal cortical circuitry can drive decision making that can result in compulsive drug taking or not, the inability to disengage from it. We also know that what's interesting is that in long-term drug abuse, the cortex itself undergoes significant changes and remodeling. So prelimbic cortex activity and excitability can decrease. And you can actually rescue this effect with stimulation, and that'll reduce compulsive drug taking. Right? And so we also see that infralimbic cortex can get completely dysregulated depending on whether you're in abstinence or in active drug taking, and similarly across a number of different areas of the cortex. So what was interesting here is this is looking at the percent density of DBH, which is a marker for norepinephrine and dopamine actually, but for norepinephrine in this case, fibers in the cortex. And these are male rats that are on the left here, blue, and then chlorpyrifos in green, pink controls, and then chlorpyrifos in green, again. And what you can see is just a general pattern across these different regions of the cortex is that it appears that females are much more substantially affected by alteration, by this gestational day 6 through 20 exposure to chlorpyrifos. And what we found, what was really interesting for us, is this dichotomy between the prelimbic cortex and the infralimbic cortex, where you get a substantial reduction in DBH fiber density in the prelimbic and an increase in the infralimbic. And this starts to actually approximate what a drug-addicted brain looks like after long-term use. So we see this in rodents, where rats that have been taking cocaine. Rats that have been taking cocaine for about a month will start to have very similar phenotypes for DBH fiber density. So we really thought that this could be, very interesting in terms of, well, okay, so we have a pre-addicted brain almost here, like, the phenotype similar to a pre-addicted brain, and now we're going to throw them into an addiction paradigm and see what they do. And so we really think that, a lot of these excitability changes, which are altered by the neurotransmitter norepinephrine, and since these are occurring in females, we primarily are going to look at females for the rest of this talk, because the changes that we observe are in females, and I think it money sorts with the males at this point. So to start out, I'm just going to remind you guys we're using the seeking persistence paradigm. These animals are going to get their compound Q, FR1. They're going to undergo extinction, forced abstinence, and then they're going to continue on with their seeking persistence extinction 2 weeks later. And so to start out, we have cocaine self-administration here. In green, this is chlorpyrifos. Pink is the vehicle, the safflower oil vehicle. And what you can see is that overall, what happens during just self-administration and acquisition is that you get a large-scale increase in the amount of variability in the groups. Overall, there's a significant effect for female rats to have increased seeking and self-administration during the self-admin when they've been administered CPF during development. And we thought this was really interesting because they aren't necessarily actually getting as more drug overall, but they are taking a much higher amount of seeking behavior to try to acquire the drug during the task. And this is although there's a timeout period, right? There's a 20-second timeout period in which they can't get the drug. So what we think is kind of starting to happen here is almost a perceptive response, right? And what's interesting is that when we continue on and we look at extinction to day 1 responding, so now no more drug, no more lights, no more tones, they actually seem to have a lower extinction day 1 magnitude of responding. So this is kind of interesting because they're suggesting that, okay, on extinction day 1, they're not going to work as hard to try to get the drug. And that this response is actually persistent. They still are not going to look for the drug. But what was interesting that we found was that if you look at their extinction rates, the number of days it takes them to actually fully extinguish, to no longer seek the drug, it takes the females that were exposed to chlorpyrifos quite a bit longer. So we're thinking about a perseverative model where maybe perhaps not in an individual instance, in an acute testing scenario, but over a long period of time, they perseverate in their adaptation to the design, right? And when we think about what is important in terms of addiction, the question isn't necessarily how much they take the drug or how much they want the drug, but how long are they going to keep trying to get the drug for? How much is it going to take for them to stop? Because it doesn't really matter if they take a ton of drug and then they just stop the next day. That's just, a binge is a lot less dangerous than continued rapid cycling response. So we think that at this point, our hypothesis for this is that CPF-exposed animals fail to show long-term disengagement from compulsive drug taking. So we have an increased cocaine-seeking persistence in duration, but not necessarily magnitude. So we wanted to follow up and say, does this have anything to do with general motivation? So we then did the same exact thing in a separate group of animals for sucrose. So this is showing our self-administration again here for cocaine. We did the exact same paradigm, but with sucrose. Pink is, again, the vehicle-administered animals. Green is the chlorpyrifos administered animals. These are active lever presses. Sucrose active lever presses are quite a bit more than cocaine. They get basically a food pellet each time they hit the lever. Everything else is essentially the exact same thing. And what's interesting is that if you look at active lever pressing behavior, they don't seem to really be nearly as interested in sucrose as the controls. And this is something that you very rarely observe in animals that aren't considered to be extensively depressed. So if you do learn helplessness or you stress out animals, repeated restraint stress, they might stop taking as much sucrose, right? Now, that is after an extended period of time. If you do it acutely, they'll ramp up their sucrose sugar intake quite a bit. And so we were interested if their motivation for sucrose was going to be altered, and so we did Sucrose Seeking, so we took away the sucrose. And similar to that, the cocaine, right, we don't see much active lever pressing behavior occurring here. And if we have them work harder for each pellet, so we go from FR1 to FR5 to FR10, and so on and so forth, they don't seem to elevate their actual amount of effort required for each pellet. And this isn't just a satiety issue because it's only 20 pellets here, 40 pellets here, whereas they're getting 200 normally. So we basically see that they are less motivated for sucrose in general. And so we have this question that starts to emerge here of whether or not they're showing an adaptive or a maladaptive response, right? I mean, you could look on the surface and say that it's probably not great to eat a bunch of sugar, right? But at the same time, there's a normative degree of necessity for sugar in the environment. Rats do not often get sweets, right? So when they have this, the caloric value of it is quite a bit higher and it's adaptive for them to take it. Now, when you have rats having free administration of sucrose along with their other food, it's actually much more difficult than you would think to get them to get overweight. You have to have, high-fat diets that basically have a poor caloric detection method. But if you give them free sucrose without binging, they don't tend to actually gain that much more weight. So there is this, so this should be a normative response that when this is binging available, right, they would want to take on as much as possible. So we became quite, curious in terms of how do we start to think about what an adaptive or maladaptive response would look like in a rodent that is not showing normative behavioral responses, right? So we wanted to look through a number of other different kinds of scenarios. So we wanted to look at auditory sensation seeking, which is basically they are in this operant chamber, the same one as before, but they're just not given cocaine. They've never been in the operant chamber before. They're literally just hitting the lever to get tones. And it's considered a perseverative response. So why would they do this? They should habituate to it and be like, nothing really is happening here. It's playing off their natural investigative nature. So they want to investigate it, say, ooh, what is that, right? But then after a while, it doesn't do anything, so they'll stop. We then also looked at what's called the elevator plus maze to look at maybe anxiety-like behaviors. We used a locomotor task to look at norepinephrine sensitivity. So is there a question of whether or not their noradrenergic sensitivity is changing over time? And then we also wanted to know, based on that general anhedonia data, right, are they just generally depressed? Or do they have depressive-like symptomatology? So overall, we hypothesized that across these tasks, maybe we would have something that would suggest that the females have a decreased ability to habituate to the task when presented, or disengage from tasks that might be a negative stimuli, like for swim tests, where they have to be suspended in water, right? And the adaptive scenario is to struggle up until a point. So to start out, we'll look at sensation-seeking behavior. So it's a measure of repetitive, non-habituated behavior, as I just mentioned. This active lever, just like before, right, produces a tone, but no drugs or alternative rewards, no sucrose, and these are naive animals. They have not done the sucrose seeking or the cocaine seeking behavior. The inactive lever, which I haven't really mentioned much before, but it does nothing, as the name would say. And then, so here we're looking at tone hits on the active and hits on the inactive lever. That's what these different bars are. So it's going to be the number of tone pairs that they get, the number of active lever presses they hit, and the inactive lever presses that they're doing. So basically, overall, you can see that all of these measures are elevated in the CPF-administered female rats. They seem to be essentially just continuously banging away at that lever. And what's interesting is, like, much like the self-administration, they're hitting the lever a lot during the timeout period, which is why the tone measurement and the active lever pressing measurement is not identical. So similar to this drug seeking, so they have this perseverative behavior where they're kind of like rapidly hitting on this lever and not necessarily adapting as much. Now, that would be fine if this tone measurement was identical to the female seroadministered vehicle. However, because it's different, that starts to suggest that they are, in fact, not habituating as rapidly or not learning that this lever does nothing nearly as quickly. So, we see that this repetitive non-habituation occurs, indicating possibly that they have a lack of a disengage mechanism. And just to note, disengage mechanism, as I mentioned before, is something that is driven by cortical norepinephrine. The ability to disengage or differentiate between tasks has to do with a norepinephrine tone in the cortex. Whether this is also on a U-shaped curve. Too much or too little will put you in either direction. So we looked at elevated plus maze next. Basically, this open arm, you can see it right here. It has no walls. It's brightly lit. The rat should find it aversive. The closed arms are a safe zone. They're dark. They like to hang out in there. But basically, this, once again, is feeding off of the natural desire for a rat to investigate its surroundings. And so they'll pop out into the center compartment, kind of peek around the edge, run back in or run across. But they're going to want to know what's out here. So all rats, unless we put them into a separate intervention, most normative rats will investigate these open arms for a brief period of time, maybe 100 seconds out of this 500-second period. However, when they've been given CPF, what you see is that they spend quite a bit more time out in the open arms than do their vehicle-administered counterparts. Obviously, closed arm tend thus would be less time. But one of the interesting things that we saw was that it seems to be a very minimal differentiation with these rats between the open, closed, and center time. So the center time is how much time they spend actually decision-making to determine whether or not they're going to go in one of these other compartments. And so this lack of differentiation of, should I investigate or am I safe, is also kind of a maladaptive response, right? So you could suggest that this is just saying that they have a decrease in anxiety-like behaviors, which is absolutely possible. But at the same time, they seem to also just not differentiate between the compartments. So is their decision-making process not really occurring, or are they just simply walking back and forth without stopping to pause and determine which area they're actually going into? And when you have this kind of like similar number of seconds spent in each compartment, it starts to indicate that they're essentially spending the entire time, which they are, just walking around without any regard of pausing to determine which compartment to go into. So we think that at this point that this is also somewhat of a, in females, I should say females, not males. This should be showing, that this is like telling us that there's a more maladaptive kind of response occurring here. So we next wanted to know, we wanted to look at locomotor behavior and just see if they had increased motor responses, which this is looking at distance traveled, centimeters over a 2-hour period. When they're administered vehicle on the test day, Chlorpyrifos, developmentally exposed Chlorpyrifos animals have an increased motor response compared to the sapphire blood vehicle animals. This response is, this could be one of the reasons they have perseverative behaviors, just an increased motor response, absolutely, which would also tie into increased noradrenergic signaling in the cortex as well. What's interesting is that clonidine, which is an alpha-1 agonist, so this is regulating negative feedback mechanisms. So if you have an alpha-1 agonist, you actually have decreased signaling. And clonidine is known for its sedative effects, so we gave them 10 mg/kg clonidine, which should knock them out pretty well. Not complete sedative effects, but should decrease their motor behavior substantially. And what we found was that the chlorpyrifos administered animals had a significantly higher efficacy of clonidine, suggesting that the alpha system may be more responsive in the CPF administered animals than in the non-CPF administered animals. We also tested with ST91, which is an alpha-2 adrenergic agonist that doesn't cross the blood-brain barrier, to try to determine whether or not this was an effect that was observed just centrally or also in the periphery. And we seem to think that this is just primarily a central effect, as the efficacy was not significantly different with the ST91. And finally, we looked at the effects of chlorpyrifos on the force swim test. And in this task, they are put into this jar of water where they can't touch the ground if they're standing, and they can't escape from it. And so rats will float. They'll just, they're not going to drown. So it's basically a good way to check out whether or not they're going to, give up. So the way that this task was primarily validated was by using SSRIs in rodents and seeing that you could actually get decreased struggling in this, or increased struggling, decreased mobility in this task, and that would be then this is a good way to suggest that you have decreased depressive-like behaviors. Over time, however, we've started to reinterpret some of these results because they're in a scenario for 15 minutes where they can't escape. It's a room temperature water scenario, so it's not super aversive, but it's not great. They don't like to be suspended there. So at some point, you would expect during that 15 minutes that they would pause to catch their breath, right? Because if they're extending their for the entire 15 minutes, if they're moving for the entire 15 minutes, if a chance to escape presents itself, they're not going to have the energy to engage in it. So then normatively, we think about this as, less than a third of the time spent is spent immobile. Most of the time, 2/3 of the time spent in most animals is spent, struggling, trying to find an escape route, swimming down to the bottom to look for an escape hatch. That's like a unique behavior for rats. They'll try to see if there's some way to get out on the bottom. And what's interesting is with the CPF animals, we see this decrease in immobility, but almost to the point where they're not spending a single second using their ability to just float and examine their scenarios. They're almost moving the entire time. So once again, we're thinking about this as, they aren't disengaging from their standard behavioral change here. They're basically just saying, over a number of different behaviors, right? Whatever it is that they're doing, they just kind of keep doing it. So this is shown, we saw this in the elevated plus maze where they're just walking around, right? We saw this in, the sports swim test where they start swimming and they don't stop. We saw this in, even just a number of kind of like just no reward basis, looking for just active lever pressing behavior, right? They seem to perseverate in their behavioral phenotype regardless of what they're doing. And for some reason, when they're asked to make some kind of motivational effort in order to engage in a reward, like cocaine or sugar, that's when they stop. So the negative behavioral response phenotype of like, do they want to work for something that's important, seems to be primarily dysregulated in them. As well, this kind of goes alongside with what we see in the cortex with norepinephrine signaling, right? Their ability to determine, or what we have found over years of research looking at norepinephrine in the cortex, is their ability to determine what's important and make a decision as to how they're going to proceed further with it seems to be massively diminished because of overactivation in some regions likely and underactivation in others. In terms of moving forward with these experiments, we think that these females show a maladaptive pattern of decision making. Some of the ways to possibly treat this would be to go through and look at, increased noradrenergic signaling, injections of norepinephrine, using the DREADS methods in order to get around this, and possibly just kind of look at, what mechanisms can we start to target in adulthood that could allow us to turn around and reverse some of these like maladaptive behaviors that are brought on by developmental exposures. So from here, what I've basically shown you is we've spent a lot of time talking about divergent neural circuits that I've shown you that contribute to differences in cocaine-seeking behaviors. In general, we saw in the normative models that increased cocaine-seeking persistence in females is driven by noradrenergic signaling primarily. We can ameliorate some of these effects by inhibiting norepinephrine signaling. We extended this to say that developmental chlorpyrifos, which may alter noradrenergic systems, we show that it does in fact alter noradrenergic fiber expression in the cortex of adult female rats, and that this occurs in a region-specific manner. we see that there's a number of different papers that suggest that decreased neurogenic fibers in the prelimbic cortex in particular may alter decision-making behavior, to alter compulsive drug taking, either long-term, as it reflects a neural state observed following long-term cocaine abuse. In our behavioral models, we showed that developmental CPF altered disengaged mechanisms primarily in a variety of tasks that may indicate a production of like an addiction-prone neural phenotype. And what I mean by this is not just a magnitude of expression, meaning like how many times they hit the lever per se for the drug, but also their inability to no longer hit the lever when the drug context is no longer presenting with it. So this idea of changing the state. Are they more prone to then getting into a cycle of abuse, because even if they've been off the drug for a long period of time, that trigger may still be present. Similarly, their behavioral profile that we saw with decrease in mobility in forced room tasks, lack of differentiation, EPM, may suggest that they have maladaptive responses when asked to disengage from a behavior. So when we apply these things then to our models of what drug abuse is, this can then impact a number of different stages. So we have drug addiction, acquisition, the feel-good stages. The normative behavior would be to disengage when this escalates, right? So we know that our data would suggest that developmental CPF could then actually alter this disengage mechanism and continue on to drug escalation. Even once drugs have escalated, the use to soothe and negative life consequences occur, the normative behavior would still be, once it starts to have too great a cost, would be to disengage again. But once again, if this disengage mechanism is disrupted, this is lost. And so generally speaking, we also have this question of like, what is the role of norepinephrine at each of these stages? This increased investigative behavior, this escalation of active lever pressing early on, the seeking of drugs early on, may be impacted by increased noradrenergic transmission. And then similarly, as noradrenergic transmission increases later on, you get the adverse consequences of drug use become less impactful. So overall, we want to conclude that environmental toxicants, such as chlorpyrifos, could induce neuroanatomical phenotypes, in our case for chlorpyrifos, alter noradrenergic signaling, that can significantly impact decision-making, addiction, and other behavioral phenotypes. And with that, I would just like to thank my lab, primarily my postdoctoral mentor, Gary Austin Jones, my collaborators, Ron Hart in particular, for a lot of this work for the K99, my amazing undergraduate students that I've had over the years, and of course my funding sources, including my K99 and the T32 from this department. And of course the rats. All right. Thank you all.
Questions for Amy? Dr. Buckley?
So, very interesting presentation, and I have multiple questions, but
Okay.
I'll focus just on the one.
Okay.
How do you control, in terms of behavioral observations, precipitation, all of the other can't quite explain, well, maybe you can explain behaviorally, for the additional acetylcholine that they're also experiencing, okay? Because I could see that the additional nervous muscle twitch energy may manifest itself in some of those precipitative behaviors.
Absolutely, absolutely. I do think that it's a major component of what we're seeing with that. I think that in terms of, so when I was showing you guys what the noradrenergic fiber density was in the cortex looked like, one of the things that I mentioned was that the males didn't seem to have this effect, right? And so when I think about what's happening in males, I think that that might be primarily driven by an acetylcholine effect, ignoring the norepinephrine effects, and their behavioral responses are somewhat different. They're still altered in a way, but their direction is different. And so what I think I'm going to have to do in terms of addressing that is look at comparative anatomy with the acetylcholine between the males and females, and see if this altered norepinephrine in females, because that system seems to be just generally more sensitive in females, is what's contributing for then that accentuated effect in females. I think that's more so where those conclusions would lie. But I think that absolutely, I mean, there's increased motor effects. Acetylcholine can absolutely do that, right? I think that there might be, what would be interesting is to look at possibly like, almost like a seizure model, see if I can push their seizure sensitivity, and that would be a great way to look at also whether or not acetylcholine is going to be largely, you know, independent of norepinephrine, is going to be necessarily altered as well. I think that's a really great point.
What would happen if you gender altered the animals?
Oh, that's a good question. I think that would be really cool. So to use like the what is the XXYs or
Right.
yeah, absolutely. I think that so I think that a lot of what is happening in the brain in terms of the noradrenergic pathway is actually hormone dependent. So not necessarily the XXY, but then doing the you know, testosterone exposure during development or something like that would be really interesting. One of the things I didn't show, we do see essentially like really aberrant progesterone, testosterone, estradiol levels in these animals at PND21. It's actually rescued at 60, so they're normal at 60, but like females have like 4-fold higher testosterone levels at PND21 when they're exposed to chlorpyrifos than normal. And so there is also this question of whether or not pubertal actions might be coming into play. In some of our other studies, we've also shown the role of progesterone and estradiol in driving up and down respectively, or down and up respectively, motivation for drug-seeking behavior. And so I think that hormones have play a much more like understated but strong role in driving a lot of these behavioral phenotypes.
One more kind of question. I haven't given a lot of thought to disengagement.
Mm-hmm.
That's kind of a new concept to me, or newly espoused, although well established in the literature. I'm always concerned when people refer to animals as making decisions. You know, as if this is a conscious, reasoned out thing versus an anatomically driven one. And I don't know which is correct.
So I guess it depends on whether or not you're talking to somebody that works with mice or rats. And the reason I say this is that a mouse, if you take a Barnes maze, a radial iron maze, a prime maze, right? You drop a mouse in, whatever direction they're facing, that's the direction they're going in, right? And you take a rat and you put them into the same maze, and they'll stop and look around. And you can actually measure norepinephrine signaling in the LC, or rather LC firing, when they're actively making a decision in like a 2 alternative force choice task. So we do know that even in rodents, and that we can actually alter that, right? We can excite the system more and have them just kind of make more mistakes, right, in choice tasks. Or we can, you know, have them emit completely. If we go even further, they'll just shut down and pause completely and just freeze up. And somebody the other way, right? So, I mean, the question is for decision making, when you think about this in terms of rodents, you do have to think about your model. I don't know personally, maybe this is my bias towards rats in general, that mice make decisions. But I do think that they're, you know, based on firing rates, based on the idea that, okay, we know that before they choose left or right, let's just say that, before they go left or right, that the LC fires, and then they do their task, we can start to suggest that same kind of thing occurs also when you're looking at humans, when they're actively making a decision. We can start to address whether or not they're choosing something. But you know, what did you choose? I don't know if we can necessarily ask them to reflect on that. I think that's more of a question of, that's where it becomes more difficult. Can
That's
we
sort of teleologic.
Yeah, absolutely.
Dr. Smith?
Are there differences in the noradrenergic fiber density in the prelimbar cortex in humans too, between sexes?
Yeah, absolutely. So it's one of the major sexually differentiated regions of the brain, is the LC. And so because the LC is so different, is so sexually dimorphic, that yes, you do actually have differences in fiber density throughout the cortex and in the hippocampus coming from the LC. The LC is actually larger in females and more densely populated of neurons, and has a higher ratio of CRF receptors on it, so it functions at a higher rate for stress, which can cause, you know, and we see this both in human women and in female rodents, this, you know, stress responsivity of chronic versus acute stressors, right? Acute stressors seem to have a much more maladaptive impact in females, but chronic stress, in comparison, chronic stressors seem to be more well adaptable. And I think that this has to do with this idea that if you have a greater baseline innervation, your stress curve, right, changes. How to push yourself. Remember how I mentioned a second ago about how if you have too much norepinephrine, like they just freeze, the animal just completely freezes, right? So that curve shifts, right, based on how much baseline is present. And that's kind of one of the points I want to make about these animals, these CPF animals, having this increased noradrenergic tone, right? Their baseline has shifted to something else. And so their behavioral profile has now shifted because of that as well. But yes, to specifically answer your question, yes, females, humans do have similar
And
differences.
it's also hormone driven, I guess?
You know, I don't know, actually. So I think, so the TH gene is an imprinted gene too, so I don't know actually where that comes from, because you would think that if it was so genomic imprinting is, you know, you only inherit from your maternal or paternal allele. And so I'm, you know, it's curious to me that if you have a gene that's imprinted, wouldn't it not have a sex different associative you know, target? But I don't know. I'm not really sure actually whether that's hormone driven or not. I'd have to look into that.
We'll turn to our audience that's remote as well. We'll start with Dr.
Yeah, I've got a question that was kind of like more speculative. So I've heard that genetics kind of play a role in addiction. Have you come across any of that in your literature? And kind of like a two-part question, would you think that some of these changes like norepinephrine and the fiber density would be consistent in some of the offspring? I know it's purely speculative, yeah.
So one of the things that I am looking at as part of my K99 is transcripts in the hippocampus that are involved, that correlate to increased drug seeking or decreased drug seeking. I'm calling them resilience and susceptibility transcripts, right? Resilience in males, susceptibility in females. And I think that in terms of, you know, are there polymorphisms for alcohol? Yeah, absolutely. I don't know off the top of my head how that relates necessarily to neuroadrenergic innervation of the cortex. That's not something that I know. Would I expect that polymorphisms in, you know, genes that are regulating beta adrenergic receptors or alpha adrenergic receptors might in fact then contribute to addiction phenotypes? I would predict that most likely they would. Could I tell you what direction they go in? Not necessarily, because once again, it has to do with not just this yes or no question of do you have more or less, but also where do you fall on the curve. And I think that for humans in particular, it becomes a much bigger question because our own life experiences, right, will also influence where we fall on that curve in terms of like stress reactivity. So it's a very interesting question. I will absolutely look into whether or not there are particular polymorphisms or transcripts that might be involved there. I think it's a lot easier to take a look in runs. And it's definitely a plan to look at particularly like how these, how different gene cascades are being expressed to contribute to this.
Great. Let's also turn over to our audience on the WebEx. I know there's some questions in the chat box. We have that as an option. Dr. Laskin can mediate and ask those questions, or anybody can unmute themselves to ask their question.
It's organized house is intro. So I'm asking, like on day 16, did you do vaginal smear and consider the ovary cycle ultrasound of the animals?
So I have done, okay, yes. So I have done vaginal cytosis on these animals and looked at cycle phase differences. The seeking persistence paradigm is not affected by cycle phase, and so for that I did not do it here, and I did not get enough variability in the behavioral results so then to actually split these guys up by like whether or not they were in estrus. I tested them at any phase for these particular results. And that's all based on my 2017 paper where I show that the seeking persistence paradigm is unaffected by cycle phase. And we think that's due to the fact that on ED1, they actually get a 4-fold increase in corticosterone levels, which kind of circumvents any effects of progesterone that you might see. Now, that's not necessarily the case for the other paradigms that we look at. So one of my other projects looks at motivation for drugs of abuse, and we actually see that progesterone and estradiol play large roles across the estrous cycle phase in determining how much motivation they have to get drug. But in this particular study, we did not see effects of estrous cycle phase.
Gotcha, thank you.
Hi, this is Debbie. I see there was a message from Kim. She's still on?
Can you hear me?
Yeah, we can hear you, Kim.
Okay. Great talk. Amy, why do you think there was no difference in center time with the EPM monitoring measure communicating, but there was a difference in any fiber PFP sub areas between chlorpyrifos and control? And also, how are you operationalizing struggling behavior in FFP? Are you calling that climbing?
Yeah, climbing I'm considering part of struggling. I just merged it for that, to start off on that one. I didn't merge swimming, because swimming behavior is a diving mechanism. I did not merge that, so I just put that separate. For your first question of the let me see if I get this correct. So you're saying the amount of time they spent in the center was not different from controls, which it wasn't. And that in terms of their PFC, I don't know. So one of the problems that I'm having with that is that, okay, their center time measure is not different, but their closed and open arm time measures are different. And so I'm trying to figure out, and I have to go back and look at the data, is I'm going to be measuring it as a velocity measure, because one of the problems is that I think that that's just by chance, because they're literally running around the maze, like as though the thing is a box and the lights are off. It's really strange to observe, because most of the time when you look at an Elevated Plus maze, like, they really just are inching, right? They're not going anywhere. And then they're like peeking,
yeah,
and like, yeah, so I think the reason it's all like, baseline is that they're just treating it like it's an open field with the lights off. So it's, so yeah, that is anti-anxiety like, but it also is just kind of not normative at all, and not what you would expect them to be doing. So I think that I think that actually Brian's point about the acetylcholine and the fact their motor behavior is a little bit higher might be something that is playing a role in that more so than necessarily the noradrenergic system. One way to test that would be to actually, I was thinking about this, is to give them a mild sedative, and put them back in the EPM and see if I could get them to get into a state of decision making to see if decision making is actually altered. Or, I don't know, run them on a wheel or something for an hour beforehand, because they're really excitable.
Is there another metric that might, like, number of passes?
Oh, yeah.
Not just time in the open, could number of passes distinguish one group from the other?
Yeah. So number of entries is definitely one of them. I didn't have it up there, but it's quite a bit higher. And
okay.
It's
so there's your explanation.
Yeah, absolutely. They're definitely going higher. I think that the velocity measure is one thing that we are gonna. I have all the videos, so it's cool because it'll tell us, basically, you can compare it then to their velocity in the open field and say, okay, are they really identifying or changing their behavioral pattern once they give them the chance to move around outside of their home cage. That answers that.
Okay. Cool.
Thanks,
and also, I disagree with the ambassador's brand. So, all right, thanks for your answer.
Thanks.
Thank you, Kim.
This is Debbie again. I have a simpler question. You showed us all this pretty data with Chlorpyrifos. Do other drugs of abuse cause, or similar drugs cause similar effects? How generalizable is this?
Ooh, I see.
Other things that would increase the acetylcholine.
Yeah, I would think that, yeah. I mean, in terms of, so one of the things that I want to just kind of make a point about this is that it's not Chlorpyrifos itself that's super important here. It's that we can do an environmental toxicant and we can develop a profile based on that. And we can form a hypothesis based on what we know the actions to be to say that it might be more effective in other respects. The question here would be, I think for Chlorpyrifos specifically, would be whether or not it would generalize to opiates, which is a question that we're absolutely interested in pursuing. And similarly, would be an action like, a DC-71 or something like this, or another toxicant or NPTP or something else that's also very well established. Whether that would have the same exact effects, I don't think that it would have the same effects, but I still think that it would be very interesting to develop a profile, given the amount of interest in drug abuse right now, as well as escalating levels of drug abuse, as to whether or not the environmental toxicants that we have present induce any kind of profile for drug-seeking behavior. I think that a lot of this in terms of getting future interest in this type of work will probably mean a move from something like cocaine to an opiate-based model. Unfortunately, there's a little bit less known about the noradrenergic system in particular in those models. But in terms of generalizability, I think that the idea would be the take-home is that we can develop these kinds of models here. And that the effects, I guess, from other acetylcholine cholinergic agents would probably be similar. But, given that the system seems to be a lot more sensitive than I even predicted it was going to be, my initial prediction with this experiment was just that maybe we would get one or two behaviors that would be altered by this, substantially. Because there didn't seem to be that much in the literature that would indicate otherwise. And so just seeing how intense this can be, you know, it'd be really cool to play with much lower doses. And see kind of like how do these profiles start to build depending on what toxicological agents we're, you know, allowing in the environment.
Sure.
Yeah.
So the other thing, just so you may not be aware of, is chlorpyrifos is on the chemical threat list. It's considered an agent of concern by the government, and therefore one could think about expanding interest in that or investigating that in terms of development of countermeasures, but using your behavioral endpoints.
Cool. Yeah.
A lot of funding for the counteract program.
Yeah, absolutely. I think
just submitted the grant an hour ago, I can tell you.
Yeah, that sounds great. I mean, yeah, it can definitely, you know, we are able to do a lot of different behavioral assays here and neuroanatomical assays. And yeah, it'd be pretty cool to say, you know, what behavioral effects a number of different threatless kind of compounds have. Yeah.
We'll have another discussion about that.
Yeah.
That's great. Thank you.
Thank you.
Any other questions from the WebEx?
I don't see any online.
Okay. If not, please everyone join me in thanking Amy once more.
Thank you. Excellent presentation.
Thank you.
All right.